I'm interested in how RNA is regulated. So RNA is this really important mediator of gene expression in the cell, but it also has a lot of different functions as non-coding RNAs. What I'm specifically interested in is how RNA regulation is important for health, but also how when RNAs are not regulated properly, they cause disease. The community here and just how great the faculty here are in terms of their mentorship and collegiality. My PhD project was looking at how a huge group of RNA viruses that include like dengue viruses, West Nile virus, Zika virus, how all of these viruses interfere with the way the cell degrades RNA, and that's thought to cause disease. So from those experiences, I recognize that studying RNA can really tell us a lot about how diseases actually work and what goes wrong in our bodies to make us sick. It has huge potential to unlock a lot of new therapies and diagnostic strategies for diseases that we really have no cure for. So there's a whole bunch of neurodegenerative diseases that are a really big problem for our society, and we don't have cures for them, or really even very good treatments. So the idea that you can target RNA processes that seem to be in common among all these different neurodegenerative diseases as new therapies is kind of what I'm hoping to contribute to in the future, and what I think makes RNA a really exciting topic to study. One thing I want to study is how the molecular machinery that dictates RNA function also is regulating the degradation of RNA, the formation of RNA, and the localization of RNA in the cell. But secondly, I'm really interested in a whole group of genetic diseases that can cause neurodegeneration and other diseases, and they're caused by defects in RNA. So trying to make model systems to study how defects in RNA cause neurodegeneration is another major aim of what I'll be working on. One of the aspects of the field that will advance, of course, is new technologies. So figuring out how to make RNAs and manipulate them and study them in the cell and in an entire organism. Another really important thing that I think will result in the future is figuring out how RNAs are dysregulated in disease. So what can we actually target in the cell to cure someone of a neurodegenerative disease? Could that be a therapy made of RNA, or could it be a therapy that targets RNA regulation? I think these are things that will probably become more commonplace as time goes on. The RNA Biomedicine Center has casts a really wide net over the entire university. That includes faculty that have expertise in these next generation sequencing technologies, so we can measure the degradation rates of all the RNAs in the entire cell. And the person who developed one of those methods is actually here. We have people that study RNAs at the single molecule level in living cells. So Nils Walters actually does that in his lab, and he is directing a core facility that's all set up just to do that specific technique, which is quite rare, actually. But there's also a lot of other people that I'm interested in working with here that are more focused on disease and pathology, like how do diseases work in animal models, for example. And I'm more of a molecular biologist, so I'm really excited to have the chance to work with people who work with animal models and can directly work with patients. So one of the great things about University of Michigan is that we have a medical school that's integrated right into the university. And there's already systems set up like the RNA Center that allow you to collaborate really cross-disciplinary to all work together towards one goal.